Are original biomolecules preserved in dinosaur specimens? That's a question that this particular article asks and presents evidence for endogenous, meaning original, collagen in Edmontosaurus fossil bone. And this is I was going to say it's an important paper, but it's important from the perspective of the sort of creation science discussion. All right, young Earth creationism. That's why this is an episode of Critiquing Creationism. Why do I say that? Because the authors, or at least a couple of the authors of this particular paper, the ones that I know are instrumental in pushing this particular research, are young Earth creationists. Right? And young Earth creationists have been saying for a long time that evidence of original biomolecules in dinosaur bones, which are 65 million years or older, just defy expectations and therefore are evidence of a young Earth. Only dinosaur bones that are really 4,000 years old and trapped in a global flood could have collagen left in them. At least that's what they say. All right. And so now they've published an article in a secular journal showing the presence of collagen in fossil bones. How significant is this? Is this a significant paper? Is this something that, as you might imagine, many young Earth creationists are going to be referring to this article over and over and over again? They're going to say, look, young Earth creationists are doing real research. They got published in secular journals. They are providing evidence that this collagen exists in dinosaur bones. Case closed. The Earth must be young. All right, wait a second. Back up. All right, is this a surprising result? Is this an earth-shattering discovery? Is this novel? It absolutely is a novel, right? Collagen's already been found in dinosaur bones many, many times before. In this particular article, they use a couple new techniques which allow them to peer a little bit closer into some of the collagen remains, specifically looking at hydroxyproline, which is a modified version of proline, a portion of amino acid in the proteins, which is almost unique to collagen fibers. And so finding the presence of that particular modified amino acid allows you to be able to say with some assurance that this was collagen that you're actually looking at. All right, so that adds another a level of assurance that this is real collagen and it is actual biomolecules, like carbons and hydrogens and oxygens that are from the original collagen molecules. They still might be modified to a great extent over long periods of time, but nonetheless, those original atoms are still there from the dinosaur tissue. So let me hit a couple of highlights of this paper, and then we're going to talk about how this isn't, I don't think this is really that as nearly as exciting as some young Earth creationists are going to make it out to be. So that's why I'm doing this particular episode, so you're aware of this paper and maybe have some ideas of how you might respond to it. All right, first of all, let's note a couple authors here. Brian Thomas and Stephen Taylor. Those would be the two that I know from this particular article. Stephen Taylor is the primary author, or it's the lab that this work was done in. He's the PhD advisor of Brian Thomas. Brian Thomas recently received a PhD from, let's go down to the institutions down here. Got to remind myself of where they're from because I can never remember the name. All the way down to the bottom of this paper. Here we have it. Corresponding author, Stephen Taylor, Department of Electrical Engineering and Electronics. All right, so publishing in this analytical chemistry journal, we have a member of the Department of Electrical Engineering and Electronics from the University of Liverpool. And Brian Thomas recently received his PhD from Liverpool. Brian Thomas is an employee, all right, a full-time employee of the Institute for Creation Research in Texas, all right, a young Earth creationist ministry. And he now is Dr. Brian Thomas. I've known him just as Brian Thomas for a long time. He's worked for ICR for a long time. Here he is noted as being a member of the Department of Electrical Engineering and Electronics at the University of Liverpool. That's not illegitimate to say that. You don't have to reveal like your employer. At the time that he would have been writing this paper, he was finishing up his PhD. And so he was an active member of, as a graduate student of that particular department. So I don't have a beef with the listing there and not acknowledging that he's a member of ICR. Stephen Taylor is very active in young Earth creationist circles as well, in his private life, not in his life in terms of his job. And so that's what's notable about this paper, is that these are two young Earth creationists who they took on this research for the express purpose of trying to show that there is endogenous material in these dinosaur bones, because to them, this is a great confirmation of a young Earth. Right? They don't believe that collagen could have survived for 65 plus million years. I mean, this paper is, in fact, making some headlines. Right? I just searched Google News for dinosaur collagen a few moments ago. Oh, by the way, this paper from MIT chemists explained why dinosaur collagen may have survived for millions of years. Fascinating article. I actually reviewed that not too long ago, talking about all the different molecular mechanisms for preserving those original biomolecules in place. And then we've got this. Okay, so here we go. The Jerusalem Post picked up this article by Stephen Taylor and Brian Thomas. Collagen found in dinosaur fossil challenges long-held beliefs. That's a typical clickbait title from the Jerusalem Post. Nature. This is not about this article, and I was blown away when I saw this particular link. Let me skip it for just a moment. I'll come right back to it. Science alert. Beyond doubt. Proteins in fossil from actual dinosaur, claim scientists. Collagen discovered in 70 million-year-old dinosaur fossil. Right? 66 million-year-old duck-billed dinosaur bones reveal decayed collagen proteins. Dinosaurs. Groundbreaking revelation settles 30-year-old debate. The other suggesting there's been a debate for 30 years. Has collagen, actually like original endogenous collagen, still preserved in dinosaur bones? Are we really debating that? Is that a debate? That's in Newsweek, right? So this little article here really has found its way into the popular press and made some waves. Now, let me go back to this Nature article right here. I did not know about this until I looked this up just 15, 20 minutes ago. Taphonomic variation in vascular remains from Mesozoic non-avian dinosaurs, Scientific Reports, which is a journal from Nature. And let me show you that article. I didn't know what this was about until a few minutes ago. Then I opened it up and voila, perfect timing. Just absolutely stunning timing, given I was going to talk about dinosaur preservation of collagen in dinosaurs. And then I come to discover that just a few days ago, right, just a couple days ago in Scientific Reports in Nature, I missed it, taphonomic variation in vascular remains from Mesozoic non-avian dinosaurs. And look who the first author is here, Mary Schweitzer. Mary Schweitzer, who's like the one that sort of began the whole soft tissue preservation and dinosaur thing 30 years ago, right? One of those headlines, 30-year debate. Does this solve this debate? Here we have Mary Schweitzer, who publishes a couple days later than this other article, with this whole article coming from Nature, a prestigious journal, right, with co-authors, looking at a bunch of different dinosaurs and examining a couple different hypotheses, testing various hypotheses about the preservation of tissues in there, and specifically the preservation of vessel structures, or some of the material that might form vessels, which would be some collagen. And what's the last line of this particular abstract? The results from this in-depth, multifaceted study present strong support for endogeneity of the fossil-derived vessels. In other words, yes, there is material that makes up the wall of large vessels in bone, all right, preserved in bone, do seem to have endogenous biomolecules. But they also detected evidence of invasive microorganisms, which apparently had invaded and taken up residence on the surface of this material. Which leads to questions about what we see in electron microscopy and so forth, what's endogenous and what isn't. And I'll just scroll down here so you can see so here we have electron microscopy of vessels that have been extracted from dinosaur bones and from ostriches. I mean, can you tell the difference? I can't just on first look. So lots of stuff, lots of staining, a ton of work here. All right, a whole lot of labor going on to produce this particular paper. I haven't had a chance to really look at it yet, but I need to read this paper. So I am informed as to what Mary Schweitzer has done recently, because she always does really cool work. Okay, I was going to say, let's come back to the paper and take a look at it. But you know what? I don't want to save the really good stuff to the very end. I tend to do that. I'm just going to break it out right now. Let's break out the big guns, make my big point, the paper that I wanted to share with you. That's going to be my aha moment. You ready for it? Let's do it. And here we are. Pretty exciting, huh? I got to scroll down before you get to see what's there. All right. Well, first point is it's from the journal Nature. All right. One of the top journals in the world. All right. Just general journals of all sciences. Right. Look at the date. You see the date up here? August 6th, 1966. 1966. All right. Slightly older than me. All right. So we've got 1966 Nature. Guess what? Guess what this is going to be a report of? Okay. What do you think they're reporting? Wait for it. Wait for it. Here we go. Cells, collagen fibrils, and vessels in dinosaur bone. And you thought this was something very recent. You know, we always hear young earth creationists say, like, scientists are shocked, by this discovery. There's surprise. Look, there's 100,000 scientists, right? They study all different fields and areas. We all don't know all the literature. Only those who really are like into certain things, all right? Like paleontologists, they would know the literature. The younger creationists talk to a doctor or, somebody who's a chemist, and they're like, I found collagen. Well, the chemist doesn't know this literature, right? So they are surprised when they hear about collagen. They don't know when collagen was found or what the evidence for is or what the electron microscopy looks like or any of that type of stuff, right? Back in 1966, we already knew, all right, that there was what looks to be collagen, individual cells. This doesn't mean the original cell. I mean something that is like the characteristics or the shape of a cell is preserved in dinosaur bones, specifically osteocytes, which is what most of the discussion is about. And all the other literature after that, Mary Schweitzer's work is looking at that. So let's just read a little bit of this, right? Collagen, right? Collagen has been found in mummified human remains several thousand years old. So there's evidence that collagen clearly survives for thousands of years. And in mastodon bones preserved under permafrost conditions. Mastodons are extinct, and we find abundant collagen in their bones. And by the way, going back to the human collagen, right? Thousands of years old. I don't know how many videos I've watched of young earth creationists explaining about soft tissues and collagen, and the little and if you take collagen and you put it in a bottle and put it on, up on a shelf, it only lasts for like a year, right? So how could it last for 65 million years when it's all like degrades within a year? Right here, mummified humans that are several thousand years old still have collagen in them. So obviously, under some conditions, collagen lasts a lot longer than just a year. All right, but this continues. Studies on Pleistocene bones have demonstrated that it is also possible to observe collagen and other organic materials in a fossil bone. Pleistocene, a couple of million years old. Electron micrographs of fossil bones of a deer-like animal from the Miocene, that's 25 million years old, reveal collagen fibrils on intact walls and caniculi, right? Again, in bone. So now we're back to 25 million years old being recognized in 1966 that these things were already seen. There have also been reports of striated muscle fibers, epithelial cells, and melanophores being found in tissues of specimens from the Eocene 40 million years old. Deposits from Eastern Germany. Furthermore, electron microscopy investigations of patches of almost completely demineralized bone of a dinosaur from the Upper Triassic. So you demineralize the bone and you see what's left. That's, by the way, that's what Mary Schweitzer and others are doing, right? And they're finding out that there are these little bits and pieces remaining around the mineralized sections that have endogenous material, meaning original biomolecules. Now, when I say original biomolecule, if you've watched any of my hemoglobin challenge videos, that doesn't mean that they're in the original shape that they were in. They might be highly modified, like hemoglobin, complex molecule. But hemoglobin actually isn't found in dinosaurs, even though we'll say that blood is found. What it is, it's the breakdown product of hemoglobin that results in a really stable structure, the heme molecule. That is what's still preserved. And so it's a similar situation here. We're not saying that full-blown collagen, like the entire collagen molecules are there, just like, they're like collagen that was in you and I right now. Let's continue with that sentence. 200 million year old and dermal armor of Devonian ostracoderms have revealed a fibrillar nature structure, while a further examination of the same material showed a mixture of mineral and collagen patterns. Right? Like in this case, impressions of fibers that would have been collagen. So not necessarily the actual collagen itself, but you can tell that collagen was there. That's from the Middle Devonian, 380 million years old, has indicated that the degradation products of collagen can still be recognized. More recently, it has been demonstrated that the fossil dentine and bone-like tissues in Devonian ostracoderms can be decalcified and sectioned on a microtome. The presence of these tissues of polysaccharides as well as collagen have been indicated. Now, this is a pretty early report, and they didn't really have the biochemistry to be able to establish exactly the biochemical nature of these things, but they're noting the possibility of these original biomolecules being present. I would remind you, this is 1966, okay? We're not in 1990 at this point. And this goes on and on and on. This is basically a review of all the different places where, the lacunae were a typical shape present in normal bone, but varied according to plate by cutting. Decalcification showed cements akin to isolated osteocytes. All right, we can see the shapes of these osteocytes in bone. And so on and so forth. So here we've got the fibrils, and then down here, here's an osteocyte, right? You look at the shape of this cell, right? Now, this doesn't mean that you can pluck this out and be like, oh, it's got a plasma membrane, and I can see that it's got all these different proteins that are part of the cell. No, we don't identify those. What we see is like this shape has been held for so long, and then parts of the organic molecules here have been highly modified into highly stable organic molecules, which then play the role of taking on the shape of that cell. But it's not like it's the same original material that was on the outside of that cell or the cell wall. All right, so that's from Nature. All right, so there you go. There's Nature saying collagen has been found. So as don't fall for clickbait that, whoa, shocking, we found collagen, all right, in a dinosaur bone. Right? There's nothing shocking about it when we already knew about that in 1966, that it was present. What we've done, or what these authors have done and others, is they have, with much better techniques now, been able to show the more of the characteristics of these things. But essentially all they're doing is confirming things that were already suspected to be there. All right, so that's my big thunder, is the 1966 Nature paper, right? So that's a good paper to have in your mind when you are when you see young Earth creationists make these really bold claims. And the best young Earth creationists won't necessarily make these claims because they're more familiar with the literature, but certainly the ones on the internet and on YouTube comments and so forth, they're sure that nobody could possibly have thought that collagen would survive that long, and they'd be shocked by it. No one's shocked by that particular article, right, from 1966. And there was many different reports in the literature. What they were reporting was, here's other literature which shows the presence of these cell-like and vessel-like and collagen-like molecules or structures in bones from dinosaurs. And since they're reporting on other literature, and this is 1966, that means there's other literature from before 1966, which shows that. All right, so this is not a shocking, revelatory piece of news overturning something. Right? It may be overturning misconceptions that plenty of scientists have had. But but not ones that really understand and know this particular material. I'm not going to review this particular paper. There's nothing wrong with the work here. This is all legit work. You don't have to be a young Earth creationist or an old Earth creationist or whatever to think that this work is reasonable stuff. You know, they took pictures. There's electron micrographs of the bones. All right. They did a protein analysis with a bunch of really fancy techniques. Right. Got all kinds of fluorescence and so forth, and, you know, spectrophotometry, right? And identifying the amounts there. And we get down to, I think, the one figure, all right. Oh, sorry, one table. Average concentration of hydroxyproline. So what they specifically were looking for is, with one of the tests, was hydroxyproline. Right? Proline is an amino acid. Hydroxyproline is a slightly modified, has a hydroxy group on it and an OH group. So it's just a slightly different. It's a very, very simple molecule. All right, very simple organic molecule, which has a lot of stability already. But the neat thing about hydroxyproline is that since it's a modified amino acid, and actually, interestingly, it's modified by ascorbic acid, which is vitamin C. So having vitamin C, one of the things it does is it modifies proline, and that proline then is used in making collagen and in quite abundance, right? So quite a bit. So collagen has a high amount of this particular modified amino acid as opposed to other proteins. So if you find the presence of hydroxyproline, right, that particular bond, then that's a good sign that you've found collagen. Right? And that's what they do. And so here in table 2, we have average concentration of hydroxyproline in 5 independent experiments. And so we have the Edmontosaurus, turkey bone, and bovine. And, you know, so here you can see there's large amounts in turkey bone and bovine, right? Huge numbers, and tiny, tiny numbers for Edmontosaurus. Because that, you know, so they found it, but it's not like it's found in abundance. Here's the one thing I would say about this article. Like, you know, if I were a reviewer, actually, the reviewers for this article, they're not thinking about young Earth creationists. They're not thinking about whether or not collagen can be preserved for millions of years. They're thinking about the techniques in this paper. I mean, what's the title of the paper? I mean, what's the title of this journal? Analytical chemistry. Can you use these tools, and what can you find using those tools? Like, right, here's how these tools function. Here's what I found from it. Right? It's not really about the question itself in many ways. Right? It's a more of a technique-driven journal in which you can report your results. And then presumably somebody else will take these results and interpret them in some other broader context. Right? What's the meaning of these results? Right? And so so this is clever, right? They're not publishing in a journal where they're getting into like having to do some serious interpretation of, like, they're not having to come out and say, well, we don't really think that that this collagen can actually survive for 65 million years. It doesn't say that anywhere in this paper, right? They're just reporting what they saw. And at the end, they do have a little summary. They say, detection of soft tissues of fossil bones is a growing field of study, and this paper contributes to the list of such findings. That's true. There's many, many papers that are similar to this. This is not, you know, they use a little bit different technique, and they're studying a different bone, but there's many other papers that show the same type of results. Right. So this isn't new, shocking, great. It's a contribution. There's no doubt about it. This is a contribution to the growing body of evidence. Corroborating results from a novel combination of 3 independent analytic techniques are presented, which taken together provide experimental evidence for the conclusion that collagenous protein remnants in some dinosaur bones are original, meaning endogenous, to the fossils, and thus providing further evidence addressing the long-standing controversy in the scientific literature. There have been people who suggested that's not, that there's something else going on. I don't think there's really that much controversy about collagen, though. There's controversy about the vessels, all right? There's controversy about blood cells. There's controversy about some other claims, or DNA fragments, for that matter. Controversy about collagen, not so much, because we've always known collagen is a highly resistant molecule, that there's a lot of it, and that cross-links with itself, and under various conditions, it basically becomes an extremely stable biomolecule that can probably persist for, if it persists for 1 million years, it can very well persist for 100 million years or 500 million years, under whatever condition it is that helped it persist for the first 1 million years. All right, so what I'm saying is that they are not pushing their young Earth creationist, feelings in this paper. They don't have to, and the reviewers of this paper, I'm sure, were completely oblivious to that. Right? They had no idea that 2 of the authors are young Earth creationists, and I'm not here saying there's anything wrong with young Earth creationists doing this. They're publishing valid results. Now they're going to use this paper. Right? They're going to trumpet this paper. They're going to make a big deal about it. It's like, look at us. People say that young Earth creationists can't publish in the secular literature, or they refuse to. And look, we did it. Okay, you did it, but you're doing it in analytical chemistry. You're not publishing an article that is like developing a new hypothesis that is challenging like the traditional hypotheses about the age of these dinosaur bones or something like that. Right? You're just publishing something that's not controversial. So not too surprising that that's your end around to get published. But it makes it look like they're, doing legitimate science. And as I say those words, I am saying they are doing legitimate science. Right? This is a completely legitimate paper. I don't find anything wrong with it. However, if I were a reviewer, there was one thing that I would say that to me, it would be a real test. Like if they wanted to write a paper that actually did evaluate how long can collagen last and what would be the trends in collagen degradation, then I would have added mammoth samples, right? Or mastodon samples. I would have added samples that we say are 10,000 years old. And then I would have added some samples that we say are like 1 million years old. Right, we have like a fossil horse, or we have a horse that's preserved in glaciation, in glaciation state, which is like 45. Actually, I think it's like 950,000 years old. So find something that the secular literature says is in the range of, 500,000 to 1 million or 2 million years old, and then analyze and do your exact same methods. Right. Use your same methods and look for the hydroxyproline and look at how well that's preserved, per volume of material of collagen, because you can see here the numbers are really low, meaning that it's very degraded, right? Yeah, there are little bits and pieces that are hanging together, and they could actually find several amino acids together to get a sequence, but it's not like this is fresh collagen by any means. All right, so turkey bones, bovine, fresh stuff. They didn't find anything in between. That's what it would have been interesting, because I'm pretty sure I know what the results would be, right? If you find something that's 1 million years old, especially preserved in permafrost or something, you're going to find that there's a lot of collagen there, right? And the collagen's in pretty decent shape. You're going to get some pretty high numbers. And the thing about Stephen Taylor and Brian Thomas there is that I would put them in a little bit of a bind, okay? Because you see, any ice age material or pre-ice age material, all right, that has lots of collagen, meaning it was well preserved, it's lasted that long, is going to be a problem for young Earth creationism, because that's got to be more than 4,000 years old. But the flood is only 4,350 years ago, which means these dinosaur bones are only 4,350 years old. You understand that? That these authors, at least 2 of the authors of this paper, believe that these dinosaur bones are only 4,350 years old. So in other words, they expect to find collagen. Right? And what I'm telling you is they should expect to find a lot more collagen. Like, these numbers are very low. Because I think mastodons and a bunch of other animals they could have gotten access to that are on the hundreds to millions of years old would probably have a lot of collagen still. And since they would be almost indistinguishable in age from dinosaurs in their timeline, right? That tells them the dinosaurs should have a lot more. Right? Now, what I think is if you were to line up something that is fresh versus mummies of human beings versus 10,000 years old versus 100,000 years old versus some samples that are a couple million years old. You have to do quite a few samples under a number of different conditions because the conditions make a lot of difference in terms of the degradation. But you could probably get some kind of like curve, right? Of like, it's really good, and it's getting worse and worse and worse. It's degrading more and more and more. And then you find your dinosaur is like way down on the curve, like it's mostly degraded. But all that other stuff should be about the same age in the young Earth creationist timeline. So that would not meet their expectations. Right. And so that's why I'm a little bit like, they cheated. They really cheated in this article because what they did was they did something that's modern and they did dinosaurs. And they didn't give us anything in between to be able to give us that, they didn't want us to get that impression, right? That there's a decay curve here. They didn't want you to see the decay curve, right? They just want you to know it's there. And to them, being there is the critical thing. Right? Because in their mind, somebody believes that the world is, that these dinosaur bones are 65 million years, well, actually 130 million years old or whatever, that they believe they believe that those who believe it's that old should believe that it's completely gone. There shouldn't be any there. I don't have a problem with it being there. I know lots of people are like, it's not that shocking that there is some bits of endogenous material. You notice that there's, there aren't any other proteins that are there that are found, right? We're not finding leptin or insulin or lipids from the cell membrane. We're not finding DNA. We're not finding RNA. We're not finding the ribosomes. We're not finding. There's lots of other parts of cells, right, that we know are less stable than collagen, and they're missing completely. However, if you go to a mastodon, right, you will find lots of other proteins still intact. Right? You'll find DNA. How do I know? Because we've sequenced an entire mastodon. We've sequenced the entire genome from a mastodon that's hundreds of thousands of years old. And yet no DNA fragments in dinosaurs, which are in the young Earth timeline about the same age. Okay, so that's it. I really did this just to show you that 1966 Nature article. All right, collagen reported in multiple different reports of collagen and other cellular remains in dinosaur bones, reported in 1966. So this isn't a shocking discovery by any means. All right, let's stop there. We're done critiquing creationism for the day. Have a good one. Bye-bye.